from an exogenous peptide, a bacterium, or a virus, all right, or some other foreign cell. They then they process that with the lysosome because it came from the outside. It's considered to be exogenous, and it is attached to an MHC2 molecule. So this is MHC2. So that's going to be presented on the surface of our antigen-presenting cell. That is utilized, so any T helper cell, not just any T helper cell, but any T helper cell whose receptor matches up with that antigen and MHC2 molecules will click onto that. Otherwise, they don't match up and nothing happens. It bumps into it and moves on somewhere to some other cell. But it has to recognize both. And when that happens, we have this interaction between the CD4 and the other surface molecules and this co-stimulation. So the macrophage activates the T helper cell by making interleukin-1. And if it's a macrophage, if it's a dendritic cell, that will happen. If it's a macrophage, the T helper cell can also activate the macrophage by making it more phagocytic. Remember, macrophages had two roles. One was just general phagocytosis, and the other was as an antigen-presenting cell. The dendritic cell had only one role. It doesn't go around gobbling up pathogens. Okay? Its only role is in the antigen presentation. Yes, ma'am?
So how does the T helper cell get receptors that match to an antigen it's reversing?
For the T helper cell, that happens in the thymus, this through random gene mutation. And somehow there's enough of them that there's millions of different combinations. But every receptor on this T helper cell is identical. So these are all the same. A different T helper cell would have different matching receptors. Okay? And somehow there's a T killer cell that also has the same matching. You look at the odds of that happening together on the same virus. It's like, really, this happens? So the T helper cell, when it's active, is capable of making interleukin-2. The receptors for interleukin-2 are on the T killer cell. Interleukin meaning between white blood cells. So the interleukin-2 molecules activate the T killer cell, and now when it separates from the macrophage, it's capable, its structural changes inside, metabolic changes inside, allow it to make its own chemicals. They're not called interleukins because this is not a white blood cell. Okay? So it makes the perforin, which perforates holes, allows both water and the granzymes to go in and kill this cell. Now, I used a virus-infected cell as an example, but it could be a tumor cell, a cancer cell that the macrophage ingests. And it's because it's a cancer cell, its genes have mutated. And it's going to be presenting quote-unquote non-self antigens on its surface. And so the same thing can happen. In fact, I know we're doing the case study on leukemia. I heard on the news yesterday where, I don't know if it was Science or a medical journal, but they had a case of 5 individuals who had leukemia, and they had taken their T killer cells and trained them to recognize the surface antigens of the tumor cells, and all 5 individuals were completely cured of their leukemia. They had a 100% success rate. The treatment that the group is going to talk about focuses on a different, it's similar, but this was just came out. Okay, yes?
Not to get off topic, but what's the gamma interferon?
So that's what the T helper cell sends back to the macrophage to make it the super killing machine.
That's why it's not an interleukin, because it's not going.
Well, it is a form of interleukin, but it's called gamma interferon. But it certainly would be classified as going between. It's just not given a number. Okay, So let me just briefly talk about the B cell. We will repeat this again tomorrow. But we have, I didn't want to have to do the whole thing tomorrow, so I didn't want you to feel like I was dumping the kitchen sink on you before spring break. Okay, the bathtub then. I'm dumping the kitchen sink, but not the bathtub. So antibody-mediated immunity, what we just got through talking about is identified as cell-mediated, and that's because of the direct interaction between the T killer cell and whatever cell it's killing. With antibodies, the B lymphocyte making the antibodies could be in the lymph node, and the virus or bacteria that's being attacked somewhere else entirely in the body. All right? So it's an antibody-mediated immunity. It still, however, requires active T helper cells. Typically, a T helper cell will only do a T killer cell or a T helper cell. I mean, that's just kind of an aside. But an active T helper cell will also be required to activate the B lymphocyte. So I'm going to erase, with your permission, my T killer cell. We'll leave the T helper cell here. Actually, I'm going to erase the macrophage. So this has happened already, okay? So in our process of, oh, you want me to leave it up there? So the T helper cell, the virus infected the macrophage. It put up an MHC2 with an exogenous peptide, and the T helper cell became activated. So now we have an active T helper cell. It's already going to be, I'll put it up on the board so you'll believe me. It's already there.
Still attached.
Not attached anymore. It's separated and undergone mitosis and cloned itself. So it's already making interleukin-2 or capable of making interleukin-2.
So where are we in our notes?
You're on B cell.
You want me or T? They're two different.
Well, we already did the antigen presentation on page 78 and 80. So now we're on page 82.
Don't act like you guys aren't the only ones thinking it.
So we've activated the helper cell. Now remember, a B lymphocyte is also an antigen presenting cell. Okay? So it's going to be able to bind an exogenous antigen to an MHC2 complex. So that's what we're going to look at here. So here's our B cell. And it has on its surface B cell receptors, which are really a form of an antibody. And for this particular B cell, they're all the same, but they are specific. They only recognize one particular epitope. A different B cell will recognize a different one.
The one that activated the
The same one that activated that, yes. So let's say that this cell binds to a virus with the particular epitope of a virus. And it's a phagocytic cell. So it's going to internalize that, and it's going to take that viral protein, and just like our macrophage, it's going to digest that viral protein with lysosomal enzymes and fragment it. Those fragmented viral proteins will then bind to an MHC molecule, just like we talked about with the T helper cell. this happens here, and then we insert our MHC molecule. And this is an MHC class 2. Because it is a antigenic, it's a antigen presenting cell. Now a B lymphocyte has a nucleus, right? So it's going to have its own self markers. They don't have any part in what we're talking about here. But remember, any nucleated cell in your body has its own MHC class 1 self markers. They're not part of this process, but I'm just reminding you that they're going to be there marking that cell as your cell. So now we're presenting this MHC2 class 2 marker protein, and along comes our activated helper cell, just randomly taking a Sunday afternoon walk. And it happens to have the same type of receptor that matches the MHC class 2. It's already active. And now it's going to activate the B cell. Instead of interleukin 2, which is what it activated the T helper cell with, it's going to activate the B cell with interleukin 4 and interleukin 6. And these activate the B lymphocyte. It will now clone itself, and it will also start to make antibodies. These B cells can do so at the rate of 10 million an hour.
That's it?
So tomorrow we're going to look at the 5 different classes of antibodies and the 2 major types of immune, well, 4 types of immunity. All right? So what happens when it activates? Remember, our inactive lymphocytes look like this.